Chapter 22 of our textbook is entitled DNA Biology and Technology, and we are going to be covering the whole chapter. And we're going to answer the following questions. Pause here if you want to jot those down. So as we've seen already in our study of genetics and cell division, DNA is the genetic material that contains all of the information needed to direct all of the functions of our body. DNA stands for deoxyribonucleic acid. It's a double-stranded molecule where each strand is made of a long string of nucleotides. And a nucleotide is a molecule composed of three separate parts. A nucleotide has a sugar group, a phosphate group, and then a nitrogenous base, giving it that nucleotide name. In the case of DNA, the sugar is deoxyribose, and these are large 5-carbon sugars, and they are linked together by their phosphate groups to form the sugar-phosphate backbone of each of our strands of DNA. Those two strands together then are held together by hydrogen bonds between the nitrogenous bases of each nucleotide. And we'll see in a little bit how these base pairs form in a very orderly manner. So it's very easy to predict the structure of DNA. The two strands join together by hydrogen bonds and then twist into a double helix. So that double helix, that secondary structure, form is done by hydrogen bonding. Each nucleotide that's bonded together to form a strand of DNA will have the same sugar, deoxyribose, like we just said, and then each of those will have a phosphate group. But the thing that changes among different types of nucleotides is the nitrogenous base. And if they're nitrogenous, that means that the main component of those are going to be nitrogen. So similar to proteins, this is another reason that we as living things need a constant supply of nitrogen, because it makes up some of the very things that codes for all of our traits, our DNA. So these nitrogenous bases are what's holding the two DNA strands together. They connect the two strands by forming what we call complementary base pairs. There are only 4 nitrogenous bases found in DNA. They are called adenine, thymine, cytosine, and guanine. So these are the same 4 nitrogenous bases found in the DNA of any living thing. Humans, made of these 4 nitrogenous base pairs, plants, fungi. All living things, their DNA is made of those 4 nitrogenous base pairs connected to their nucleotides. These bases, though, can only pair up in very specific ways, and that's because of the number of hydrogen bonds that can form between them. So adenine can only pair with thymine. Whenever you see adenine, you know it can only pair with thymine. And cytosine can only pair with guanine. And likewise, guanine can only pair with cytosine. So you're never going to see adenine and guanine paired together, or thymine and cytosine paired together. Because each base can pair with only one other base, the sequence of bases on one strand of DNA can exactly predict the sequence on the other strand. So if you see an adenine on one strand, you know there's going to be a thymine in that same location on the other strand. So if we look at this sequence of nucleotides where C stands for cytosine, we know that on the complementary strand, we're going to see a guanine. A standing for adenine, we know that the complementary strand is going to have a thymine. When you see a thymine on one strand, you know the complementary strand is going to hold an adenine. Adenine pairs with thymine. Thymine pairs with adenine. Guanine pairs with, hopefully you said cytosine, adenine with thymine, and guanine with cytosine. You're going to get some practice lining up those base pairing in your lab today. So before we jump into how that DNA becomes replicated, I'll remind you that during the cell cycle, DNA replication happens during the S phase of interphase. And during interphase, all of the DNA is still contained within that nuclear membrane. So we haven't gone into mitosis or meiosis yet. And DNA is in its loose chromatin form so that it can be replicated in preparation for cell division. So DNA replication happens during the S phase of interphase. And let's look at how that happens. When DNA replication begins, the first step involves the enzyme DNA helicase. So when we see that ending ASE, we know we're dealing with an enzyme. And DNA helicase will arrive at one end of a double strand of DNA. And it's going to break the hydrogen bonds between the base pairs, which then separates the two strands of the double helix. DNA helicase travels along the length of the double strand and kind of unzips the two strands from one end to the other. And now that the two strands are separated from one another, each exposed strand will act as a template for a new complementary strand to be formed. So we know that the nucleotides, the nitrogenous bases, are exposed, and if there's an adenine hanging off, we know we can add a new nucleotide with the thymine. Then free nucleotides, so nucleotides that are floating around in the nucleus, will form complementary base pairs with their complementary bases on each of the template strands. So in this image, the nucleotide adenine is red. So anywhere on the template strand where you see an adenine, a free nucleotide containing a thymine, or orange in this case, that base will bond to it. Anywhere where there's a cytosine, seen in blue here, a free nucleotide containing a guanine, a green one, will bond to it. And as the free nucleotides assemble and bond to their complementary base pairs, another enzyme called DNA polymerase, our second enzyme there, will come along, and DNA polymerase runs along each of those strands and bonds the phosphate groups together to form the sugar phosphate backbone of the new strand. So even though those free nucleotides are lining up next to each other, we still need to create that sugar phosphate backbone, and that's the job of DNA helicase. And that is all happening as the free nucleotides in front are bonding to their base pairs. During S phase of interphase, this process of DNA replication happens to each one of our strands of DNA in the cell. So as humans, we have a total of 46 chromosomes in each cell. So at the end of DNA replication, we'll have double that amount of DNA. That is a lot going on in the nucleus. And also recall that the replicated copies of each chromosome are called chromatids and are held together by those centromeres so that they are paired up when cell division does occur. The process of DNA replication is called semi-conservative replication, and it's semi-conservative because it will result in two exact copies of DNA, but the two original strands, the ones that were used as the templates, never come back together. So each new double helix of DNA contains one strand from that original molecule, the parent strand, and one daughter strand, the one that was formed during the replication process from those free nucleotides. So why is exact replication important? Why do we focus so much energy on creating exact copies of our DNA? Well, we know that our DNA directs all of the activities of our cells by containing all of the information needed to produce specific proteins or perform specific functions. Every cell in our bodies must have all the same information in order for every cell to be able to perform those essential body functions. So for example, when we talked about hormones during the endocrine system, a hormone affects a target cell because it has a receptor protein on it or in it that binds to that hormone. That receptor protein is coded for by a specific gene in our DNA. Without that gene that can be expressed in that cell, it would not be able to recognize that hormone and it would not respond appropriately. So it's very important that each cell replicates its DNA exactly so that all cells can function as needed. And as we saw in our discussion of the reproductive system, one of the goals, the primary goal of reproduction, is to pass on genetic information, and that's done by passing on our DNA. And if it's not copied exactly, then we're not passing along our exact genetic information. So when we get to next to our discussion on evolution and change over time in organisms, we will see that some changes in DNA do occur and how they cause populations of organisms to change over time. So in our, we just looked at how DNA is replicated and why it's important to create exact copies of our genes, and we have talked a lot about DNA's role in directing the functions of a cell in our body's functions. But we haven't really looked at how exactly that happens. So how, if DNA cannot leave the nucleus, how does DNA direct all of those functions outside of the nucleus? How does it get that message out? And that happens through a process called gene expression. So the DNA inside the nucleus sends its message to the cell through another molecule also made of nucleic acids called RNA, or ribonucleic acid. And it does this through a process called transcription. The segment of DNA that codes for a certain protein is used as a template to make a new strand of RNA. And that's the process of transcription. Then RNA is able to leave the nucleus to find a ribosome, which hopefully we remember can be found either floating in the cytoplasm or on the surface of the rough ER. And when the RNA finds a ribosome, translation occurs, which translates the message from the DNA into a specific protein that gene codes for. Then the protein produced actually carries out the function that was directed by the DNA. So the two steps of transcription and translation lead to the expression of genes in the form of the protein that was created. So in order to fully express a gene, you have to have both transcription and translation occur. So let's look at a closer look at that RNA ribonucleic acid molecule that we just mentioned. Ribonucleic acid, RNA, which acts as the messenger molecule to carry out the DNA's directions, is very similar to DNA, but it has some important differences. Both DNA and RNA are nucleic acids, so they're kind of speaking the same language by communicating in that complementary base pairs attached to their linked nucleotides. But while DNA is double-stranded, RNA is always single-stranded. They both have 4 bases, and the majority of them are the same. RNA still uses adenine, guanine, and cytosine, but instead of thymine, like we saw in DNA, RNA contains uracil instead. The other 3 nucleotide bases are the same. And the base pairing rules are also the same. And the other major difference is that while the DNA that codes for our gene functions only in the nucleus, RNA functions in the cytoplasm to carry out the directions from the DNA out into the rest of the cell. There are 3 main types of RNA that we will see play a role in transcription and translation. There are actually many more types of RNA that exist, but these are the ones that we are going to focus on for this discussion. So we've already talked about messenger RNA, that carries the information from the DNA to the cytoplasm. Then we'll have ribosomal RNA, and ribosomes are the site of protein synthesis, and ribosomes are made out of proteins mixed with that ribosomal RNA. And then finally, we will have transfer RNA, or tRNA, which is responsible for bringing the correct amino acid in the correct order when we're building our protein chain. And we'll see in a little bit that tRNA, transfer RNA, is able to do this through the language of what we call codons. And a codon is a 3 base pair sequence, and each codon reads for a specific amino acid. So in the next video, we're going to focus specifically on transcription, that first step of gene expression. See you there.